So the aim of the method in our manuscript is to provide comparative analysis of signaling pathways in myeloid cells using human and mouse systems. It helps define which signaling pathway is involved downstream of selected pattern recognition receptors. So this method is simple to implement. It relies on primary cell preparations and validated reference compounds, which is essential for the elucidation of signaling mechanisms. Demonstrating the procedure will be Ratiba Touil and Adeline Unterreiner, respectively scientists and research associates in our group.
In a laminar flow hood, set out a sterile pair of scissors, a 1-liter beaker, and a plastic bag. Place the previously obtained buffy coat into the beaker and use the scissors to carefully open it. Using a 25 milliliter pipette, add 100 milliliters of PBS supplemented with 2 millimolar of EDTA. Mix by slowly pipetting up and down, and then transfer 25 milliliters of the diluted buffy coat into 6 50 milliliter conical centrifuge tubes that have each been pre-filled with 15 milliliters of polysaccharide-based density gradient. Centrifuge the tubes for 20 minutes at 800 G with moderate acceleration without the break to allow for separation of cells based on their densities. After centrifugation, 3 layers will be visible: a pellet containing red blood cells and granulocytes, an upper layer made of plasma, and a white ring containing peripheral blood mononuclear cells. Use a 10 milliliter pipette to harvest the PBMC ring.